Hello, audible?
Yes, sir. Yes, sir.
Yes. Okay. Today, now we are going to discuss about Bryophyta. So already the last class we completed entire the algae. So what is algae and how many classes is there? What type of reproduction system? What type of life cycles? Some of the members of life histories. Okay. What is the special characters of in Cyanophyte? Similarly, Xanthophyta and Bacillariophyce. So other life histories of in Phaeophyte and Rhodophyce. So and also economic importance of algae. So this is last class already completed. So next now we are going to discuss about Bryophyta. Here before going the Bryophyta, you see this is of a cryptogram plant. Means the cryptograms are divided in three categories. One it is in algae, Thylophyta, Bryophyta, and Pteridophyta. Here number two that is in Bryophyta. It is intermediate position of entire the that is cryptograms. Means in between place is there in cryptograms. After the Bryophyta, Pteridophytes, before the Bryophyta in algae is there. That's why it is intermediate positions. So some of the characters we are looking in algae character, some of the characters we are looking in pteridophytic characters. That's why this is intermediate positions in where the cryptograms groups. Okay. Yes. So now first you see here Bryophyta. The first term of Bryophyta was coined by his name is Brown, Braun, 1864. It is including algae, fungi, and lichens, mosses all together. In 1864, Brown scientists discovered by first Bryophyta. Okay, so next to Skimfer, 1879 treated Bryophyta. It is some special characteristics is there. It is divided in three categories. His name is of Skimpler, that is liverworts, hornworts, and mosses. So bryophytes are also include the simplest or most primitive members of an embryophyta. You see here, after the bryophyta, we are saying that it's in totally embryophyta. But these plants are not looking to embryo. But here some embryo type of characters are looking, and in spite of it is there is no to embryo. That's why we call this is in primitive embryophytic plants, embryophytic. So next, his name is a Tipo scientist. In 1942, he proposed the term of ataraxia of bryophytes because lack of vascular tissues is there in plant body. That's why. So here means the bryophyta plants are there is no vascular systems means there is no xylem and phloem. That's why we are saying that is in non-vascular plants. First point. Again, you see, so these bryophytes are can be okay, as see this of chlorophylls, autotrophic, embryophytic, atracheophyte, archegoniophyte, amphibian cryptograms. Okay, so these are all the names of bryophytic plants. First one is of chlorophylls, which contain the chlorophyll. Autotrophic, self preparation of food, and embryophytic. So embryo also you are observing in this one. And atracheophyte, there is no proper, and tracheids is there, means non-vascular plants is there. And archegoniae plants, so female reproductive organ, there is of archegonias. And next one that is in amphibious cryptograms. These plants are that is amphibian plants. So when these plants are going to reproduction system, it wants to the water. There is no water. These plants are not going to the reproduction systems. That's why we are saying here it is in amphibian plants. And also one of the most important point here, the generation, when you are looking the life cycles, here two types, two sections is there. So alternation generation of their life cycles. Gametophytic generation is there and sporophytic generation is there. See, before in algae, so when you are studying in the ectocarpus, from there to the sporophytic generation is there. Before the ectocarpus, there is no, so they're dominating in gametophytic, spores are only. From you see how the sporophytic is going to independent. From in ectocarpus, the sporophytic is totally, sporophytic is comes to outside the sporangiums. From here, in when you are studying in Bryophyta, so here the sporophytic, okay, alternation of generation, gametophytic generation is there, and here also we are looking in sporophytic generation also is there. This is a little bit again. You see one more some special characters of in Bryophyta. Yes, the bryophytes are a small group of primitive land plants. See, in algae, it is totally where the water, where the rainy seasons. Even though bryophytic plants are also, we are looking heavy rainy seasons where the shady places in spite of. So these are all we are looking in where the walls, where the water, where the land. Okay. So primitive land plants growing up predominantly in where it is an amphibian habitat. So individually, they are small and inconspicuous but become conspicuous with growing in tufts and cushion-like structures. In these plants are some cushion-like. So when you are looking in these bryophytic plants, some of the cushion-like structures is there. The plant takes bit of an heteromorphic alternation of generations. Gametophytic generation is there, sporophytic generation is there. That's why, so we call this is in heteromorphic alternation of generations, in which gametophyte is dominant phase, sporophyte is physically, physiologically, depending on where the gametophyte. So here this point is most important. So you see now here, when you are looking in the one plant, where it is here, I'm showing one plant of in gametophyte. So now you see here, this is all the bryophytic plant. These are all the bryophytic plants. Yeah, here now you see this one, where it is in which one, this is of in, this is of Funaria, and here now you see Polytrichum. So where the that is of in Markamsia, this of in Rexia. See, when you are looking in the Rexia plant, this is totally thallus-like structures. So from here you see when second one B, that one, this of in this is also Rexia. Yeah, here now you see this one, this is in Markamsia, third one. Okay, so this is in Rexia, round like structures. This is B also in Rexia. C it is in Markamsia. So D, this is in Porella plant, and next one this is in E, this is in Anthoceras, this is in Funaria and Polytrichum. See, so these are all the plants are in bryophytic group. These are all the plants are bryophytic group. Follow visible diagrams.
Yes, sir.
Yeah, now you see this is of in A, okay, B, both are Rixia plant. C, this is in Markamsia, Porella, and Anthoceras and Poly. All together bryophytic group, but here little variation is there, variations. That's why you see here. So here we are not looking clearly in sporophytic stage. The sporophytic is embedded in the thallus. Okay, but here now you see the sporophytes a little bit comes to outside the thallus. But when you are looking here, entire the sporophytic stage comes to outside the thallus. So here is totally comes to outside. That's why here the sporophyte is partially depending on gametophyte. You see here this one, this is all gametophyte, here this is in sporophyte. But here it is in gametophyte, here it is in sporophytic stage. This is in alternation of generations. Okay, the plant body primitive, like when you're looking in entire the bryophytic plant bodies. So some of the members, some of the plants, that is of in liverworts, thallus with rhizoids, liverworts, particularly when we are looking in, these are all the plants you see generally for C, a little bit A and B. So these are all human liver-like structures. That's why we are saying here it is in liverworts plants, liverworts. Okay, and second one, you see here this one more is there here, number E. So this is in anthoceras. So this anthoceras is in hornwarts, means the sporophyte comes to outside. So we are looking horns like structures. That's why, sir, these are all the plants are hornwarts. Yeah. So next, now you see here this one. This is in totally with this one here to gametophyte, and here it is in sporophyte. Here the sporophyte is complex structure. These are alternation of in generations. Okay. So with number one, there's of in liverworts, hornworts, and third one, this is in moss plants. Okay. The plant body, these are all the entire the plant body consists of simple parenchyma. This entire the plant body is formation by simple parenchyma, but here no vascular systems, vascular tissues. Okay, yes. So next, now you see here the reproduction systems. So it is an advanced group. That's why this is in ogamous type. Okay, ogamous type takes place in the gametophytic. Sir, where here the gametophyte is there. Here, this is a gametophytic stage. This is a gametophytic stage. Here, this is in gametophytic stage. So, this gametophytic stage produce two gametes, produce to the gametes. So, that is of in Antheridia and Archegonia. Okay. So, the reproduction system, it is in ogamous type. Ogamous type takes place where the gametophyte. The sex organs, it is multicellular and enveloped. Some of the sterile jocket layers is there. The female gametangum, that is in Archegonia. The male male that is of in anthrozoids. Okay. So, sir, these Archegonia, which appears for the first time in the Bryophyta in evolution of in plant kingdoms. Okay. Yes, this is all female reproductive organ. The female reproductive organ, that is in Archegonia. Okay. The male reproductive organ, we call that is of in spermatozoids, anthrozoids or spermatozoids. This is of in biflagellate to whiplash type of flagellate. To whiplash type of flagellate. Okay, we call that is in male is in anthrozoids, spermatozoids. Okay, and here one is most important point, the fertilizations, okay, takes place in the presence of water. There is no water, these plants are not going to the reproduction systems. Okay, that's why we are saying here it is in amphibian plants. Okay, yes. So next, yes, the fertilized egg means zygote. After the fertilization is formation in the zygote. Okay, so this zygote is retained within venter of the archegonium, and individual it is divided metatically the form of embryo, that is of exoscopically, and wall on the venter enlarges with the developing embryo to form protective, that is a multicellular envelope called of calyptra. This is a one special type of zygote is there. The embryo develops where it is in sporophyte. Here two generation is there, gametophyte, gametes, male and female, sporophytic. This is a spores, sporophyte. Okay, so which either completely liverworts, okay, sporophyte depending on gametophyte. the partially parasitic on the gametophyte where it is in mosses. So when you are going to the advanced plant in Bryophyta, so that sporophyte is going to as an independent plant. But before means primitive bryophytic plant, entire the sporophyte is totally depending on gametophyte. Okay. Yes. So this is a little bit general characters of in Bryophyta. And one more you see, when we are looking in Rexia sporophyte, there is no seta, foot and capsule is there. So when we are looking in which one that is of an intermediate position of Anthoceras, the sporophyte is divided in three categories. That is of in foot, and instead of seta, there we are looking in capsule. There we are looking meristematic zone is there, and the third one that is in so capsule is this. Similarly, when we are looking in mass, this is a little bit advanced is there. Foot, seta, capsule, it is in complex structures is there. Okay. Yes. So next, now you see here, this is a little general characters of in Bryophyta. Now we are going to discuss in classifications. So many members are classified, so many classes, but now we are studying in only three classes is there. That three is most important. Number one, that is in where it is in life cycle, that is the gametophytic and sporophytic generations, reproduction system. This is you see here classifications. So these classifications. So we are looking in only three divisions. Number one, that is in Hepaticopsida. Number two, that is in Anthoceratopsida. Number third one, that is in Bryopsida. Only three classes is there in this one. Three classes. So the Bryophyta classifications, it is in only three classes is there. One, it is in first one, that is in Hepaticopsida. Second one, that is in Anthoceratopsida, and a third one, that is in Bryopsida. Okay. So here you see how can we recognize it. Okay. What is the meaning of Hepaticopsida? Okay. Hepaticopsida, how can we identify, how can we recognize it means here the members of in Hepaticopsida, popularly known as, that is of liverworts. The thallus shows liver-shaped lobes. Entire the Hepaticopsida plants, So you are looking in human liver-like structures is there. That's why. So we are saying that is in liverworts, and entire these Hepaticopsida almost all 280 genera is there, and near about 9,500 species is there. Okay. Yes. So next, now you see here, already I told gametophytic generation is there, sporophytic generation is this. Yes. Here I am showing number one, that is in Hepaticopsida. Where it is in this one. See number C, that is in Hepaticopsida. Okay, AB, and C, this is a Marchamcia Hepaticopsida. You see here this plant, it is a human liver leafy like structures is there. Liver leafy like structures is there. Yeah. The gametophyte usually dorsiventrally thallus. This is the thallus. So these thallus, it is in dorsiventrally. Dorsiventrally means always you see 1 2 1 2 like this. Uh, Development is going to 2 sessions is this. Dorsiventrally thallus like structures is this. Okay. Yes. So next, now you see here the gametophyte. So these gametophyte is a internal structure of simple. Simple is there. So differentiated into many special type of tissues is there. Many special type of tissues is there. That is photosynthetic cells is contain several small chloroplasts and reflective of oil bodies also we are looking these gametophyte. Okay, so this gametophyte, it is a thallus, dorsiventrally leafy like structures is there, and these all the gametophyte is contains heavy photosynthetic cells is there, photosynthetic cells, and this contains the small chloroplast also we are looking in this one. Okay, yes, the sex organs are terminals. The sex organs are with the terminals. Maybe in sometimes maybe it is a dorsal surface of the thallus. The sex organs developed from a single initial cells. From here to the sexual reproductive. This one. Okay, thallus. This one here it is in thallus. Okay, dorsiventrally leafy-like structures is there. This is in a dorsal side, and other side is a ventral side. From the dorsal side, so now here is the produce of two types of rhizoids is there. So later we'll discuss how many types of rhizoids. And here now you see this mid rib is there, mid rib, and the tip of the below just tip of the branches that we are looking at notch-like structures is there. Okay, yes, the sporophyte body. So here these gametophyte, okay, produced a sporophytic body may contains chlorophyll tissues and without, okay, such tissues where this is rexia. In rexia there is no, so which one there is a chlorophyll type of the tissues. It may be simple represented by the capsule or differentiated into foot, capsule, still further into foot, seta, and capsules. Okay, so when you're looking in this one, A, where it is in A, means there is a rexia. So in rexia, there is no sporophytic body, foot, and seta, and capsules. Foot and seta capsules. From here, so we are looking in foot and seta and capsules. Next one, the orchisporangia. Okay, the archisporium is derived from the endothecium of the embryo. The sporogenous tissues used to spore mother cells and sterile cells. The sterile elaters are single called with spirally thickenings. Yes, so next one there is a the capsule lacks of sterile regions. The wall of the capsule develops of that is of an ambithiums. It has no stomata. This is also most important point. The wall may be single layered or multi-layered. It spills on drying, but not regularly in two valvules. So these are all the little general characters of hepatic opsida. You see once again, hepatic opsida, dorsventrally differentiated, dorsventral differentiated gametophytes, which are either simply thallus or with leaf-like appendages. Okay, so and are always nevertheless internal tissues are homogeneous or differentiated, and the sex organs are born terminally or developed from superficial dorsal cells. The sporophyte similar or differentiated into foot, seta, and a capsule, always of limited growth. Sporogenous cells are endothelial origin and without any columella. So this is all and Hepaticopsida. So after the Hepaticopsida, so again this Hepaticopsida four orders is there. So that is of in Spirocarpalis, Marchamcialis, Jangermonialis, and Colobralis. So these are four orders is there in Marchamcia. We'll discuss in the four orders in later. Yes. Second one, there is of Anthoceratopsida. Here I am sharing number E. This is of in Anthoceratopsida. See, what is the variation of in Anthoceras and Marchamcia? So these Marchamcia, the sporophytes not comes to outside in the area, in the outside the thallus. Okay. So here now you see this sporophyte is comes to outside the thallus, looking like a horn-like structure. That's why we are saying here it is in hornwards. Okay. Yes, the Anthoceratopsida is represented by a single order. Only single order is there. There is of Anthocerotalis. A single family is there. There is of Anthocerotaceae. The family comprises six genera, near about 300 species is there. Okay. Next, now you see here the first, sir, this gametophyte. What type of shape is a gametophyte? So this gametophyte is also dorsiventrally, okay, lobed thallus without any internal differentiation of tissues. On the ventral side of the thallus, scales are absent. Here now you see this one. When we are looking in Marchamcia, midrib is there, but here no midrib. Okay. And now you see here the scales is there, but here no the scales. That is the differentiated between Anthoceratopsida and Marchamcia. On the ventral side of the thallus, scales are absent and unicellular smooth-walled rhizoids are present. You see here, rhizoids. Okay, rhizoids means as a root-like structures is there. So, and also one is the most important, there is no air chambers, there is no air pores, but there are some special characteristics is there, that is of in mucilage cavities. Okay, so that open out on the ventral side of the thallus. Okay, each cell of the thallus has single laminate, the chloroplast with pyrenoids. So this is the most important. No other bryophytic plant not looking these type of the characters. Okay, every section, every part is contained with pyrenoids. Okay, chloroplast with pyrenoids, and the cells are also do not contain any oil bodies. So this is a little bit of variation of Marchamcia and Anthoceras. So next one, that is of so the sexual reproduction system of Anthoceras. So here also we are looking same male reproductive organ, that is in Antheridia, female reproductive organ, that is in Archegonia. Okay, so the Antheridia arise on the dorsal surface of the thallus within the cavities. Within the cavity, this is the most important here. You see, in Hepaticopsida, where the Antheridia comes to outside like this umbrella shaped, open umbrella and umbrella shaped. Particularly in Anthoceras, embedded in the thallus, means the reproductive organs is embedded inside the thallus. Okay, within the cavities, where the archegonia is completely embedded in the tissues of the thallus dorsal side, except the there's a protruding necks of the archegonias. And also here the Antheridia, it is in group of Antheridias is there, not a single group of Antheridias. The sporophyte of consists of bulbous, foot, meristematic regions, and long cylindrical capsules. Here, this now you see sporophytic plant body. Okay. In sporophytic, in all the bryophytic sporophyte, foot, seta, capsules, foot, seta, capsule, but particularly here, this one, Anthoceratopsida, foot and that is a meristematic zone and capsule is there. Long cylindrical capsule is there. The sporogenous shows of intermediate growth in intercalary meristems. So this is also advanced characters. So from here to the sporophyte is going to independent plants. Here so many meristematic zones are intercalary meristematic zones is there. Okay. So the wall of the capsule is totally photosynthetic. Yeah, next now you see here columella. So let us be showing the clearly capsule. The columella is derived from where it is of endothecium, the archisporiums. Okay, so that is of overarches the central columella and is where it is in derived from that is of amphitheciums. The archisporium gives rise the spores of it is a mother cells and a simple of compound sterile called of pseudo-elaters. This is also one of the most important. The pseudo-elaters are with or without spiral thickenings. So next one, the capsule. The capsule of tip of the means when the spores, the wall is cut, the spores are comes to outside in this area. Okay, the capsule matures from the tip of dehiscence, the gradually in two hallways with exhibits of hygroscopic of twistings. So this is all to the Anthoceratopsida general characters. So once again, we listen here to all the Anthoceratopsida general characters. The gametophyte, dorsventrally differentiated, simple, and foliage internal tissues of homogeneous. The sex organs are embedded in the gametophyte. The female sex organs formed from the superficial, and the male sex organs are hypodermal dorsal cells. The sporophyte differentiated into the capsule and foot and meristematic zones. Lower part of the capsule here that is in meristematic zones. So that the growth of the sporophyte is not limited. Sporogenous cells are that is of an amphithecal origin. Amphithecal origins, all the internal cells are forming central sterile columella. So this is all to the general characters of in Anthoceratopsida. So next now you see here after the Anthoceratopsida, one more important is the Anthoceratopsida only one class is there. So yes, so next last one that is of here now you see E, F and G. Okay, F and G that is of Funaria and Polytrichum. This is of a little bit advanced. What is advanced here? See, this is totally thallus-like structure is the thallus. So when you are looking here, little bit plant-like structure is here, plant-like. Okay, but it is not in a true plant. From here to here, you see this is of in sporophyte. From here to here, this is in gametophyte. So when we are looking at Anthoceratopsida, so here this is in gametophyte, this is in sporophyte. So here this is in gametophyte. Here the sporophytic stages is there. Here the sporophytes are embedded in the thallus. Okay, that's why see what type of special characters of in Bryopsida. Yes, so Bryopsida are commonly called as that is of moss plants. Okay, moss plants, and they are much greater in number of more widely distributed than before liverworts plant. So almost all you see here it consists near about 700, 600 to or 700 genera, 14,500, near about 15,000 species being in largest of in Bryophyta. This is an advanced group and largest Bryophytic. groups. Okay, so they constitute the dominant vegetations box where it is in alpine regions, in Antarctic regions. They occur woodlands, okay, and hedgerows, even the polluted urban areas. They occur in dark green cushions between the paving stones and other dump of where that is of in crevices. Okay. So next now, the gametophyte is distinguished as branched prostrate filamentous juvenile stage are called in protonema. This is the one special type of structures is there. Okay. So we call that as a protonema. Juvenile stage means some little bit after the fertilizations, this plant body is produced, plant body germinated, one special vegetative structures. So we call that is in juvenile stage. We call that is in protonema stage. Okay, and so after this protonema is produced to new plant that is of in a gametophore, gametophytic plant. Okay, the protonemal stage is of in transitory while the gametophore are persistent. Persistent is a permanent. Next, the gametophore consists of stem, spirally arranged leaves. It bears the sex organs, show definite apical cells in the earlier stage of their development. Yeah. So now here this is a gametophytic stage in this. So this gametophytic stage here now you see this is in Funaria. Here it is in polytrichum. This one is a rhizoids is there, stem is there, and leaves is there. Okay. Here, tip of the branches, we are looking one special type of color-like structures is there. That is of in perigonial leaves. That is in perigonial leaves. So these perigonial leaves are contains sexual reproductive organs. Sexual reproductive organs. Okay. Here now you see a little bit variation of in area and polytrichums. So next one, the sporophyte. Sporophyte generation of in the sporophyte consists of influence of moss plant. Here this is of foot. This is totally seta. From here it is in capsules. Okay. The sporophyte consists of foot, seta, and capsules. The growth of sporophyte is definite. Seta is absent with the spagnalis, and gametophytic pseudopodium arise up the sporangiums. The wall of the capsule is several layered. There is a distinct epidermis with the functional of non-functional. Stomata is there. Stomata. This is one of the advanced character. The archisporium develops from endothecium. So that is of the central columella develop from the where it is in endothecium. The archisporangium gives rise to spores alone, and the sterile cells like elaters is conspicuously absent. So elaters we are looking in before. It's Anthoceras and Marchamtia, but here so we are not looking to the elaters. So this is Bryophyta. So here this Bryophyta is a three classes is there. There is of Spagnadiae and Androideae, and there is of Braidiae. This is the three classes of general characters of Bryophyta. Okay. So once again, so brief of Bryopsida. You see now here Bryopsida, we are saying that the mosses plant, the gametophyte. Okay. So after a distinct, though usually short protonema phase is there. protonema juvenile stage. Develops erect shoots, usually radially, preserving symmetry with leaf appendages. Okay, where that is of in phyllodes and stem-like clouds of sexual organs develop from the superficial cells of the of at the top of apical of axillary meristems. The sporophyte of limited growth, usually differentiated into foot, seta, and capsules. The sporogenous cells are endothelials or amphithecial origin, but always surrounding the columellas. Okay, so the Bryophyta near about 24,000 species is there, near about 960 genera, with plus or minus. No. So this is all to the general characters of which one that is of in Bryophyta. Okay. Yes. How many classes are there in Bryophyta? Please.
Three classes.
What is the first one?
Hepaticopsida.
Very good.
One is Anthoceropsida and Bryopsida.
Bryopsida. Okay, na? Yes. So next
Yes.
now you see here. Next now distributions. Distributions of Bryophyta. So almost all Bryophyta, it is in a cosmopolitan distributions, means all over the world wise. So we are looking in Bryophyta. Okay, so alpine regions, Himalayas bending to Kashmir. Okay, east of the India branching of where it is in Burma. Almost all over the India area we are looking in bryophytes. In South India, that is of Nilagiri hills is there, and where the Telangana it is a Mahabnar. There also we are looking in bryophytic plants. Okay. Yes. So next now you see here, what is the economic importance of bryophyta? Economic importance of bryophyta. So Bryophyta is a major role playing our daily life and human life. Okay. The Bryophyta, it is a generally little economic importance is there. Some mass plants are produced, provide is the food. Okay, herbivorous of mammals, some of the birds and other animals. Okay, so other all none of them directly important in man except some species of in where that is of in sphagnum. Species of sphagnum is grow and multiply enormously. Water, thus filling the ponds and lakes with broth. So, yes, the bryophyte is of air pollution indicators. Air pollution indicators. These are all had by many research students are also. Okay. So next now you see, so that is of geologically the mosses are of considerable importance of soil forms, soil formation, soil productions, and soil conservations. They provide essential steps in the development of the soils. The mosses grow or born soils, along with the lichens grow even on dry rocks where it is in a growth of higher plants in fossils. Without bryophytic plants, never higher plants are not coming in that area. That is the most important. Okay. So this is all to the economic importance of Bryophyta. Yes. So next here I am showing there is the economic importance of this is of distributions. Okay, so distribution means it is all over the worldwide, polar regions, alpine regions, yeah, and everything area we are looking in that one. and what is the economic importance? This is economic importance. They are not giving economic importance. Yes, in spite of is okay. Now the reproduction system is already completed. Vegetative reproduction system is there, and asexual reproduction system is there. Generally, asexual reproduction system is absent in this one, but some of the members are produced with a special type of the structure that is of an asexual reproduction system series. And next, now you see here, what is the variations? What is the differentiated? Means origin of an algae, origin of bryophytes. These bryophytes are coming from where? Okay. Already I told this is intermediate positions. Okay. So now you see here the first. This is the differentiator between algae and fungi. Grow in moist and shady places as well as in water and called amphibian plant kingdom. Algae, similar character, same. These bryophytic plant are also looking in water, so algae also are looking in water. And the tissues differentiation is well developed. Here, tissues differentiation to found in only higher forms. So this is a little variation. The reproduction system where it is in ogamous type. Here isogamous is there, aniso is there, and ogamy is there. Okay. The sex organs of multicellular. Here the sex organs here it is in unicellars. The female sex organs are archegonia. The female sex organs it is in ogonias. Zygote remains enclosed in the archegonia. Zygote here it is in liberated in the plant. Gametophyte phase is dominate. Here usually sporophyte phase is dominating. This is the variation of in. Okay. So algae and bryophytes. So these are all the economic important, and also uh, these are ecological also most important, playing the most important role in environment. Okay, this is of in economic important. This is also classifications of three types of classifications is there. This one. This is a tree plant is there. Okay, so this of liverworts is there, and here it is in hornworts is there, and third one this is in moss plant is there. It is a variation of in three groups. Picture is clear?
Yes, sir.
Yes. So next now you see this is we are going to origin of bryophytes. So origin of bryophytes. So, here I'm sharing next one Markhamshire. Where it is Markhamshire? The one that I'm showing in this one. Here now you see this is in Markhamshire plant. Now we are going to discuss about in Markhamshire plant. Markhamshire plant here now. Yeah. So division, you see, Markhamshire. So Markhamshire is one of the orders of the class Hepaticopsera. So it is in well distributed, and order of comprising near about 420 species is there under 35 genera. Yeah. So well-known genera are that is in Markhamshire. Yeah. So that is already I told Markhamshire is a liver-like structures is there. So there are about 800 species and 200 genera in this group. The basis of position of Archegonia, they divide into two groups. That is of in That is of an acrogyne, where it is in of pelleia, and anacrogyne. That is two groups is there in this one. So now we are going detailed explanation of it. Markamtia. This is the plant Markam. This one here it is in Markamtia thallus. Yeah, one, two, three, four. Total in Markamtia thallus is there. The Markamtia class division, that is of Bryophyta. Class it is in Hepaticopsida. Order it is in Markamtialis. Family that is of Markamtiaceae. Genus this is in Markamtia. Yes. Now first you see the plant body of Markamtia. Okay, this is of in gametophytic stage, which is green, thick, and flat, dorsiventrally branched. Dorsiventrally, dorsal side and ventral side. Dorsiventrally leafy-like liverworts. Okay, the dorsal region includes one that is of in species known as air chambers is there. Air chambers. The air chambers of communicate with interior through where the pores is there. The ventral region is composed of compact, which one that is of colorless parenchyma. Storage tissues is there. Colorless parenchyma, storage tissues is there. Here now you see this one, this is of gametophyte. Yeah. So the plant body dorsventrally flattened and prostate dichotomously branched. The thallus is of conspicuous and may reach length of near about 10 to sometimes here you're looking 20 centimeters is there. The apex of each thallus is lobe-like notched is there. Here now you see there you are looking one. This is the thallus. Okay. Markamtia thallus is this. You see now we are this of reproductive organ. Yeah. Here this is of in gametophytic stage is there. So these are gametophytic stage. Yeah. Dorsiventrally flattened, leafy-like thallus is there, and development of dichotomous branching is there. Dorsal side it is produced to the scales, rhizoid-like structures is there. Below tip of the branches, there one we are looking a hole-like structure is there, that is in notch. Okay. Yes. So next now the close observation, when you are looking the close observation of the thallus on the dorsal surface of reveals the presence of polygonal and it is in rhomboidal areas is there. So that is of here you are looking air chambers is there. Air chambers is there. Each polygonal area has a pore its center through which the air chambers is connected with the external where it is the environment. Okay. Yes. So next now you see here one midrib is there. The midrib present in each branch of the thallus, which is marked on the dorsal surface of shows groove and a ridge of ventral side. Along the midrib is present characteristic prominent cup-shaped structures is there. Dorsal side of the thallus. The cups are called, that is of an asexual reproductive system is there. So generally the bryophytic asexual reproductive system is absent. So here you are looking one cup-like structure is there. Gemma cups. Okay, these are in asexual reproductive organs. Okay. Yes. So next now the mature thallus are produced in sex organs. Male sex organs, second is of female sex organs. So already you know very well the male sex organ that is of antheridia. Now you see here this is of an antheridia. You're looking like this umbrella shape, that is in antheridia. So here it is in archegonias. Just 1 minute. Yes. Okay. This is of gametophytic phase is there. Yeah. This one, this of in gametophytic stage. This is in gametophytic stage is this. Dorsiventrally leafy-like structures is there. Yeah. Yeah. Next, now we are going to the internal structures of in thallus. Internal structures of thallus. So when you are going to the sectional cutting of thallus, Markamtia, here generally three regions is there. Three regions. So one it is in epidermis is there. Here it is in the sectional cutting of this thallus. Three regions. So one it is in epidermis. You know your upper epidermis. Here this of lower epidermis is there. Okay. In between upper and lower epidermis. So here we are looking photosynthetic zone is this. Third one, the here it is in storage tissues. So generally, the Markamtia thallus, it is divided in three regions. One it is in epidermis. Second one that is in photosynthetic zones, and third one that is in storage regions. Storage regions. So these epidermis are two types. One it is in upper epidermis, second that is of in lower epidermis. So upper epidermis, okay, it is a single layer. There is no any intracellular spaces in between the epidermis. So there we are looking one special type of the structures is there, that is of in which one that is of in, yeah, this one, this is a clear. Okay. Upper epidermis, lower epidermis. Yeah. So this is of an assimilatory zone. Here it is in total parenchyma, storage tissues is this. So this is of a single layer. There is no any intracellular spaces is there. So in between of these epidermis, there we are looking in one special type of the structure is there. We call that is of in air pores. Air pores. What is the main function of air pore? Exchange of the gas. So below the epidermis, above the storage tissues. So we are seeing one special type of the sections is there, that is of in assimilatory cells rich in the chloroplast. So here we are looking in chloroplast is there. The spongy tissues is there. In between of spongy tissues, their gap is there. We call that is of an air chambers is there. Air chambers. So this is of photosynthetic zone is there. Photosynthetic zones. Okay. Now you see here this of an upper photosynthetic zone is there. This of a storage. Storage means this one is a storage tissues. So storage tissues are contain different type of the food materials. Okay, that is of in starch, oils. So whatever it may be, you see here storage tissues. It below the air chambers, it more thicker center towards which one that it's confined. Mucilage. Its cells are processes, reticulate techniques, lower cells of zones here. Okay, so this is of in section cutting of thallus. Three regions is this, and also lower epidermis. You see now here we are looking two type of the scales is there. So one it is in tubercular rhizoids, branches like structures is there, and next one here it is in smooth rhizoid structures is there. So this is total Markhamshire thallus section cuttings. Yeah. So that is of external morphology, and this is in internal structure of in Markhamshire. Yeah. So next now you see here reproduction system of Markhamshire. So generally the reproduction system here, the asexual reproduction system are absent, but in Markhamshire, so there we are looking one special type of the structure is there, we call that is in Jamma. Okay, you see here by the Jamma. Now you see here this is in Markhamshire thallus. So in this Markhamshire thallus, just tip of the below tip of the branches. So there we are looking one cup-like structures is there, cup-like structures. The first one of the cell is divided two parts. One it is in basal cell, upper cell that is of in upper cell is there. Second one is upper cells. The basal cell is attached to where the thallus, and upper cell it is going to metatic divisions. How much of metatic divisions you are looking, that much of development you are looking in that area. Okay. So here Jamma cup-like structure, one stalk-like structure is there. Now you see here this one. This is of in stalk-like structure is there, and above cup-like structure we call that as Jamma cups. Here we are seeing that Jamma cups like that. Here now you see this one is Jamma cups. So after the development, when it is cut the mother gametophyte, automatically it is produced to the new plant. Okay. So these Jamma cups are germinated only at the dorsal side. These are the Markhamshire Jamma cups. Okay. Yes. So next now you see here reproduction systems. Reproduction system is there, and one more is there in Markhamshire. One where that is of an adventitious branches is there, root-like structures is there. Okay. So and next one is of fragmentations. So this is also asexual reproduction systems. Yeah, but important in only Jamma cups are important. Okay. Yes. So next now you see here this one. Here this is in development of in Jamma cups. Okay. See how the development of in Jamma. This is of in first one section divided in two parts. Next 2 to 4, 4 to 8. After the 8, here now you see 1, 2, 3, 4, 5. So these have been totally Jamma cups are developed. So next now you see here the second part. So what is the second part? That is in sexual reproduction system of Markhamshire. Okay. Yes. So here the sexual reproduction system, it is in male, that is in Antheridia. Now you see this is in male Antheridia, and female that is in Archegonia. Always, okay, na? The male it is in open, so male it is in umbrella shaped like structures is this. Okay, na? So now you see here Archegonia, they're looking like this. So this is a male gametophyte. Here it is in female gametophyte is this. So here this is the thallus. This is the thallus. This is the thallus. How the development of a male gametophyte? So now here this is a male gametophyte. Yeah. Yes. So first now you see here. Now this is the development of a gametophyte. For example, now you see here this is in thallus for number A. This is in thallus. For the initial cell is there. One of the cell, one of the sections. Okay. One of the sections, one of the cell is going to modified as a antheridial initial cells. So these antheridial initial cells are going to develop as a antheridial branch. Yes, here now first now you see the single superficial cells near growing the point of loop and antheridial disc. Functions of antheridial initial cells are this one. Here it is initial cells. So these initial cells are divided in mitatically. Okay, na? So it produced and divides by the transfer divisions from the basal cell and outer cell. Means here this one. Okay, na? Sir, this one is divided in two parts. One it is in basal cell. This one here it is in outer cells. It is divided in two sections. Basal cell. Okay, it is always attached to where the it is always attached to where the thallus. And what is the main function of basal cells? It is taking food material from the thallus. From the thallus. Okay. Yes. The outer cell divided metatically. Okay. It's the outer cell. Outer cell divided metatically. It is a formation of it is in antheridia. Antheridia. Okay. So whereas the basal cells, it is embedded in the thallus. Yes. So next now you see here the outer cells means the cell is divided two transfer divisions of it is a formation 1 2 3 4. Okay, na? Four celled filament. The upper two cells of this filament functions of primary antheridial cells and the lower two primary stalk cells is this. Yes, so next row, the primary stalk cells divided in two short two cell is thick, which one there is a stalk cells is there. The primary antheridial cells divided into vertical walls at right angles to each other, thus forming 8 cells. These cells are arranged in two tiers of 4 cells in each. So this is the development of in antheridias. So now you see here the first is divided into 2 to 4, 4 to 8. Okay, some of the this 4 it is modified as a stalk-like structures is there. You see now here after upper cell is modified as a jacket layer-like structures is this. Okay, it is epidermis-like structures is this. This is stalk-like structure. That's why in all the bryophytic male reproductive organ, it is in club shaped. It is in club shaped. Okay. So now this is of mature in antheridia. From here to A, B, C, D, and E, F, and okay, this is a development of. Sir, here it is in a stalk-like structure is there, and this one, this is of antheridia. Yes, sir. Antheridia contains which one that is of androgonial mother cell. This is of androgonial mother cell. Sir, here these androgonial cells are modified as a male reproductive organs. After the development, this area, this jacket layer of destroy, jacket area that has been destroyed, the spores are comes to outside the antheridial chambers. Okay, antheridial chambers. So when comes to outside in this area, so here, so we are looking two flagellas, blepharoplast is there. Okay, blepharoplast. So one it is of cross like structures is there. Okay, so that is of blepharoplast. Next, now you see here second one that is of in archegoniophore. Archegoniophore almost all similar development, but this of in female reproductive organ. Now you see here this is of in female reproductive organs. Okay, yes. So first one that is of in A. Here also we are looking same one initial cells first. Okay, so which contain that type of that sexual reproductive organ genes, that cell is going to divide it, that is going to is the initial which is of mother cells. Sir, here you see one of the cell first. Okay, one of the cell is going to modified as a female reproductive organ. So this is we call that is in archegonial initial cells. So here these archegonial cells are divided. These archegonial cells are divided in two equal parts. Same. One it is in basal cell, second one that is in upper cells. So they are also same, but structurally different. Structurally different. So here this is in female reproductive organ, that is in male reproductive organs. Yes, so now basal cell is attached to where the base is taking food material from the thallus. And upper cell is divided metaphysically. Okay, na? These are two divisions is there. After the two, so again it is going to divide it in this much of in layers. Okay, so how the taking food material from the thallus, that much of development we are looking in that area. After three, again it is now you see here four, where it is an upper one cell is there, we call that is in primary cover cell is there. Primary cover cell is there. Okay, you see next after this one. So here now you see we are looking one means here one important point is there. So when we are looking in entire the female reproductive organ, sir, here this one, this is of venter canal cells is there, and neck canal cells is there, cover cells is there. This three is most important. That's why. So here this one, from when we are coming here, so this below part is modified as a venter canal cells. So these upper cell is modified as a neck canal cells. Upper cell is modified as a neck canal cells, and these upper one it is in modified as a cover cells. Okay, so here this is of in development you are looking in this one, and you see when you are looking in this area, so it is modified. This is a female reproductive organ. That's why here now you see primary axial cells, means the egg-like structures is there, egg-like structures. So this is of all female reproductive organs. So okay, now you see here this is of in J, this is a development. So here it is in total female reproductive organs. So here this of inside formation by this of in egg canal cells. Here it is in venter canal cells is there, and here this one it is, here it is in neck canal cells is there. So this is the development of in archegonia in Markamsia. You see from A only single cell is a little bit, you see how this modified I going to development, and here entire the female reproductive organs. Okay, so next you see here after the male reproductive organs, here the spores are comes to outside in this area, so these spores are going to, okay, going to inside the female reproductive organs. So when entered inside the female reproductive organ, so we call that is in fertilizations. Okay, here the fertilization means takes place. So now here the fertilization takes place when the male and female thallus grow in near and each others. Water, it is essential. So if there is no water, these plants are not going to the reproduction systems. Okay. So the neck of the archegonia is directed upward on the dorsal surface of the disc of the archegonia 4. The mature archegonia, the venter canal cells and neck canal cells of disintegrate and from the mucilaginous masses. So automatically the spores entered inside the female reproductive organs. After the fertilizations, okay, after the fertilizations, here the formation it is in zygote. Okay, so this zygote is produced to the new sporophytic plant. The zygote is produced to the new sporophytic plant. Here you see now here this one, this of in a sporophytic phase. Yeah, here the sporophytic stage already I told. Okay, So the zygote charges in the size of fills, and the winter, the stalk of Archegonia begins to elongate it and becomes 3 to 4 centimeter long. These cells of the venter also divided of periclinally and from 2 or 3 layered of calyptra, okay, which surrounds the developing sporangia. A ring of cells at the base of the venter divide two form of collar-like growth known as often, that is perigoniums or pseudo, that is often perianth, pseudo, that is often perianth. See here this one, not here this one here. Okay. Yes. So this after this sporangia, when comes to outside in this area, there is the sporophytic. That I'm sharing now. See here, this one, this is in sporophytic stage of in Markamsia. So these sporophyte stage, it is divided in these are diagrams. Yes, this one. Okay. So here, this is a sporophytic stage. Okay. So these sporophytic stage are divided in three. One is a foot and a seta, and next one that is of in capsules. Okay. Yes. So next one you see life histories of in Markamsia means the female reproductive organs. The Markamsia, it is a dioecious plant is there. One it is in male, and second one that is in female is this. The male is produced to the antheridiophore. Antheridiophore is produced to the antheridium, and female produced to the archegoniophore, that is of an archegonia. It is produced to the egg. It is produced to the egg. Anthrozoids both together formation is that is of an embryo. So these are embryo. It is produced as sporangiums. After the sporangium, that is of a capsule. That is in spore mother cells. So that is of an after completed meiosis. Next, the spores are produced. The spores are produced to the Markamsia. So this is the life history of in Markamsias. Okay. So is it clear?
Yes, sir.
So this is of in Markamsia life cycles. So just this is the most important in Markamsia. Yeah. And next one. So second one that is of in Pelleya. Pelleya. Pelleya is not there in this one. Alex, this is of in Markamsia. Where it is in of Pelleya. Younger Monyeris. Just wait. That is a younger Monyeris. Yeah. Yes. So one more important topic is there. That is of younger Monyeris. Okay. Pelleya is Pelleya Florella. Okay, here the slide is not available in this one in spite of. So we are telling of Pelleya, and one is the more of the younger Monialis. So how can we identify in younger Monialis? You see, so almost all these younger Monialis also we are saying that is of in leafy liverworts. Okay, it is the largest order of the liverworts, contrast with thyroid liverworts such as Markhamsia. And here now you see this one is of in Pellia. This is the most important in our topic is there. So this is also moss plant. This is also in liverworts plant. The Jungermonialis are the leafy thyroid liverworts as the gametophyte of these area of false nature. The sporophyte is differentiated same. Know the differentiated between Markhamsia and Pilia, both are same, foot and capsule and seta. So which may be elongated. What is the importance of it? How can we identify it now? How can we recognize in Pilia? That one is enough. Pilia is so the younger monial is here about 8,000 species of 200 genera is there. Okay, so on the basis of position, Archegonia they divide into two groups. There is of anacrogyne and acrogans. Pelea belongs to that is of anacrogynes. Anacrogyne, Jangan Manualis. This is the most important. What is the Jangan Manualis are the largest order of Hepaticopsida. About 244 species is there. 244 genera is there, near about 9,000 species is there. The plant body is the thallus of leafy axis. Rhizoids are smooth-walled. Scales are absent. This is most important. Scales are absent. What is the difference between Markhamsia and Pilia means? The antheridia is a globose and subglobose is there. The jacket of archegonial neck in 5 vertical rows of cells and as broad as the venter. The jacket of capsule is 2 or more cells in thickness. The spores mother cells are become deeply, that is a deeply found, deeply four-lobed before the first nuclear divisions. Archegonia is gives rise to spores and that is of elaters also. The sporogenous with foot, seta, and capsules. Seta is much that is elongated maturity. The maturity capsule divisions by 4 valvulas. Okay, so here this is of in 3 suborders is there in that one, but in our topic only Pelea. Okay, the Pelea is of in Anacrogynous genus of thyroid form of the family that is of in Pelliaceae. That is of Gengermonialis group. There are about 4 species. Okay, the 3 are available in India. So rest of 3 plants are available in our India. The plant body, Pellia, it is a dorsiventrally prostrate, dichotomously branched with a little bit some margin-like structures is this. Dorsal surface is very smooth, thick at midrib. The thallus of terminates into notch, where the growing point is situated on the ventral surface, smooth, that is of unicellular, present below the midrib. Scales are some that is of in ecdyrozoids are absent, and so only smooth scales are is there. So next one that is in internal structures of Phallea. The internal structure of gametophyte is a simple, mainly consists of in which one that is of parenchyma cells, midrib made up of many cells or thickness is there, marginal regions and a single cells in thickness, the cells of marginal regions and the upper cells of midrib with chloroplast. Okay. So here this is often Pelea internal structures. So here next one, the reproduction system, vegetative reproduction system is there, and next one, that is often sexual reproduction system is this. So vegetative reproduction systems, already we know very well, break the portion automatically in the plant is there. So some adventitious branches also we are looking in that there, and here the sexual reproduction system is, these plants are, that is often monoecious, and next one, that is often dioecious. Same, okay, the sexual reproduction systems, monoecious and dioecious is there. The sex organs are formed in two or more rows in the dorsal side of midrib. Antheridia are born at the dorsal surface of the thallus, along with where it is of midrib. Their position is indicated by that the production, and each projection marks the antheridial cavity in which single antheridia is present. This is to the antheridia. The development almost all are antheridia. Number one, divided in two parts, basal cell and upper cells. That is a development of an antheridia. Similarly, second one, that is of an archegonia. It is also same. Archegonia is a formed group of two or, this is important point, two or 12 at anterior end of the branch. The group of archegonia is protected by invalid care. Group of archegonia formation in this one. So above the archegonias, we are looking for protection layer. We call this a invalid care. So this is the special type of structures in Pelea female reproductive organs. Okay. So after the female reproductive organs, the male comes to outside in that area. The male is goes toward the female reproductive organs. After the fertilization, after the fertilizations, okay, out of the fertilization formation, the zygote, so this zygote is produced to the new plant, that is in sporophyte. Okay, so sporophyte, the zygote secretes the wall and begins increase in size. The cell wall of the winter grows and develops the calyptra, which covers the young sporangiums. Sporangium, foot, seta, capsules. Okay. So the capsule with fully matured seta elongated and ruptures the calyptra and carry the capsule in air. The capsule wall splits into four valves at the dehiscence line and exposing the which one, that is of eletophores, which holds the spores and elaters for some time, and then the spores of, that is of inflicted out of short distance. The spores comes to outside in that area. Okay, so when comes to outside in that area, so it is taking food material from the hygroscopic, and that spores are produced to the new plant. So that is of in a Jungar Monnier list. Okay. So next we will discuss in Anthoceros and Spagnalis. We'll discuss in the next classes. So until then, thank you very much. Hmm? Yeah. ఉమ్. నిన్ననే నాకు అయిపోయిందా సార్ క్లాసు? ఇప్పుడు కాదు సార్, ఈ వీడు బీఆర్ఓ పోదా, ఇది అట్లే ఉంటుందా సార్ ఇంతసేపు అయినా కూడా? అంతే కదా, వాళ్ళు పెట్టేసిపోతారు, నేను క్లోజ్ చేసేసుకోవాలి, ఇంకా పోతలేదు ఏంటంటే నేనే తీసేస్తున్నాయి మీటింగ్ అని